subgroups, our working groups got put together effectively. So you all are in a work group now. Sorry, I misnumbered some of them at the start. So those of you who've gotten an updated number of your work group, that's the correct number. When we get the Kaizen game activated, which will be before the end of this week, I have to actually need a little more hand-holding to really pull the trigger on making it work and get you the right instructions. You can change the name of your team if you want to personalize yourselves to make yourselves a more dominant person to be competing against in our competition through the question and answer phase of the course. And you'll have to let me help you do that because I think I'm the program manager, so I'll have to do the renaming of any of the teams that are out there. And I'll leave it up to the captains of the teams to speak on your behalf and to gather any information they want about whether you want to change your name. Otherwise, today's starts the beginning of our module that has to do with how to actually get research done here at UAB, starting off with what you need to know. This is really the session on ethical conduct of research. And the first part of today's session is an overview of responsible conduct of research type training. The first speaker is Dr. Lisa Schwiebert. She is Associate Dean. Her name changes all the time, but she now is Associate Dean for both graduate and postdoctoral trainee career development, and she's also in charge of the Responsible Conduct of Research program here at UAB. One of the other things I think I've mentioned already is that she really spearheads a terrific activity that will happen in early April called UAB Mentoring Week. And we'll be providing more information about that as we get closer to the time. It's a series of 4 or 5 events, depending on how much you want to step outside your comfort zone into learning about mentoring from perspectives of different people. We'll include 2 particularly noteworthy things. One is there'll be a keynote address by a speaker from the NIH, and there will also be a presentation by an interactive theater group called Theater Delta. That's really a terrific opportunity to see acted out an aspect of the mentoring relationship. This year, the theme is going to be on trainee mental health and issues around that. And you'll see, I think it'll be a very rich program, and I encourage everybody to make time to attend. It's an hour to 2 hours each of 5 days in a row during that week of mentoring week. Okay, so I won't take up any more of Dr. Schreiber's time. Please remember that we have a lot of people participating online, and so the more I can hand you the microphone when you have a comment, the better. And I'll try to be close so you can do that easily.
Thank you, David, and thank you for the shameless plug for Mentoring Week, all things mentoring. We can chat about that at the end. Again, my name is Lisa Schwiebert, and we're a bunch of different hats here today. I've got my RCR training coordinator hat on. So how many have maybe heard one of my sessions or attended the 717? This will be a little bit of an overview, so apologies. Don't give away all my secrets. But as we go through, please feel free to interrupt, to ask questions, comments, and so forth. And apologies ahead of time if I mess up on the technology. That's why I have teenagers. All right, so as mentioned, this morning is really a refresher, if you will, of sorts. We're going to dive into a few different topics, again, an overview of all things RCR. But toward the end of the hour, I really want to focus on 2 different topics. That's data management and authorship, which really is a thread. They're both threads that run through no matter what research you do, whether it's basic, clinical, or certainly translational. So integrity, a matter of trust. I know you're all chewing and probably not awake yet. What does integrity mean to you? And for those who have been in my talks before, you know I am not going to go any further until someone says to me, making sure you're awake. What does it mean to you?
Being able to connect with what
sleep at night when what you're doing. Perfect. So really doing that right thing when no one is looking and making your conscience calm so that you can sleep. Not tweaking the data, not maybe making something up or intentionally adjusting data, but really doing that right thing when no one's looking. So really this is a matter of public trust. Certainly the public supports our science through their investment, i.e. taxes. Tax day is not too far around the corner, but they certainly also participate in our trials and give it themselves in that way. But importantly too, they rely upon us to make their lives better. And despite the current climate of angst, I'll leave it at that, really the majority of the public do support public research and the funding of it. So certainly if anything goes sideways, and many, I'm sure as you know, reports come out in the news that scientists have been dishonest or unethical in some way, that really erodes the trust that they have in us. I'd like to relay a conversation that I had several years back now when the administration was just changing hands. I do participate in 2 different study sections at the NIH, and so I ping-pong back and forth on that lovely direct flight from here to DCA at Rigor National Airport. And happened to be with one of my colleagues, and we found ourselves with one of our elected senators here in Alabama, and we're asking him, how will the change in administration really impact NIH funding or research funding on the whole? He says, what does it matter? Y'all make up the data. Right here, our elected officials. So we're up against a challenge. So we really need to maintain that trust. Certainly that extends to experimental design, unbiased record keeping, again, doing the right thing when no one's looking, the appropriate data analysis to select that right type of analysis to not just give you the answer that you want, but to really give you as close as to a glimpse of what's going on. Certainly how we collect and store data is of consequence. If it's not stored correctly, we could lose it. And we'll talk a little bit about rigor and reproducibility, and that I know it was referred to as the Kaizen game. So that plays into that topic as well. And certainly being able to publish our work to the public. There are 9 essential areas of conduct as posted on the ORI. That's the Office of Research Integrity website, which is housed on the NIH platform. Some of the examples that I'll be taking or talking about today were from the ORI platform. Definitely, if you want to do a deeper dive, there are city modules that are housed here at UAB. City, you may be familiar with, does the IRB training. They also have a course for RCR. It's quite extensive, these particular topics as well as several others. But you do get then a really full breadth of training in the different areas. So what is research? We're talking about misconduct or correct conduct, responsible conduct. But what is research?
An investigation.
An investigation, exactly. An investigation to really develop and add to our knowledge base, to really extend or move the needle in a particular area that we are exploring. So why do we do it? Why do we do research other than because it's really fun and maybe they'll give us money to do it?
To understand more or investigate a little bit more certain topics.
Sorry.
To understand or a certain topic, what influences or what are some of the things that impacts a certain topic.
Impact, that's the key, right? So we generate data to generate knowledge to have impact. As I mentioned, the public is really entrusting us to make their lives better, to move that needle, whether it's improving medications or improving medical devices or improving roads, like malfunction junction. That's another area of research, but really to have impact on our everyday lives. So that's why it's so important that we do it responsibly. What is research misconduct? Take a guess.
A lapse in integrity.
A lapse in integrity, okay. Can you drill down and be a little more specific? Has anyone heard of the acronym FFP? Feel like Ferris Bueller. Anyone? Anyone? Yes.
Falsify. Second half. Plagiarize.
Exactly. Fabricate. So falsification, fabrication, and plagiarism. And I put up these definitions across the board for the different societies, if you will, or areas of interest. The ORI, as I mentioned, and certainly the National Endowment for the Humanities, and certainly UAB, because they are all shared. FFP is that classic definition of misconduct. So fabrication, falsification, or plagiarism. What is fabrication? Creating, just sitting down and writing a novel, right? How about falsification? Changing the data, tweaking it a little bit, maybe throwing out that data point, maybe not calibrating an instrument completely correct because you kind of want that number. Well, what's plagiarism?
Yeah. Other people's ideas as your own.
Exactly. Other people's ideas as your own, without proper citation. So certainly this dribbles over into a variety of misconduct practices. Manipulating images, kind of tweaking, falsifying, maybe creating, making it up. Using inappropriate research methods that could be harmful or dangerous, or maybe they're not, maybe the incorrect analytical tool to analyze the data. Certainly poor research design or letting your bias creep in to how you design the experiments. Intentional errors, whether they're computation or analytical. Certainly poor mentoring is a breach of conduct. Making sure, I know we're all always mentees and mentors. So as you look forward to training your own trainees, but certainly as you have been trained, to have proper supervision and give guidance to them. It's really violation of human subject protocols. So yes, FFP is that classic definition, but there's really a breadth to that in terms of practices. Okay, here's a quiz. What's wrong? I don't know if I have a pointer here. But you see in the upper left, so there's something wrong with each of these figures. What do you see for the upper left? Oh, sorry. They did. So they narrowed the focus and then blew it up a little bit and increased or enhanced that image. How about the one next to it? With the multiple panels of the wild type M1, M2, M3, M4. What do you see? Which ones? Wow, you're good. So that is the same figure, except transposed like this. Just kind of turned over. About bottom left, figure 3, Western blot. Say again?
Half a band.
They're missing half a band, but what else do you see? The scale is different. Good pickup. What else?
Not showing the whole blot.
Not showing the whole blot. That's probably a pretty fair representation, though. It's flipped. Exactly right. And the last one on the lower right, the gel shift, figure 4. What do you see? He's giving me hand signals back there.
Are those lines going straight down?
They are. What do you think that means?
5, 6, they spliced it.
Yeah, they spliced it. So lanes 1, 4, and 5, exactly the same. So here's the key. And this is all too on the ORI website that I mentioned earlier. Here you can see the comment assay. Yep, they lightened it and expanded it a little bit. As we talked about, figure 2, what is that, 1 and 4 are flipped. Figure 3, right? Those 2 are flipped. And then for the gel shift, 1, 4, and 5 are the same. So this really does happen. There have been reports in the New York Times. You never want to be published, I don't think, in the New York Times. There are some incredible examples that they show on their interactive app or website of figuring out these types of misconduct, if you will. ORI has incredible tools to detect misconduct in this way. Pretty sophisticated imaging that they can detect nearly anything. So they will catch it, as do editors, publication editors or reviewers. Eagle Eye now, and really are on the lookout. So that covers kind of the imaging or manipulating data that way, but there's also scientific writing misconduct. Certainly we talked about plagiarism, but then also not including conflicting data or information from the literature that really speaks against your hypothesis, kind of ignoring that, if that's misconduct. Certainly manipulating the information or taking it out of context, or presenting data, numbers, in a way that your target audience may not fully understand. What do I mean by that? So here's an example. Item C and item A, which is bigger?
This is one of those things where you tell us they're the same size.
No, that's my implicit bias talk, but good catch. Which is bigger?
Probably
I'm going to go D.
We're not looking at D, we're looking at A and C. So you're right, there was a misleading pie chart. A is bigger, but the way that it was presented, it appeared that C was. So you can see there when it's flat on. Thing two, maybe this is more difficult to see, but the legends on the left, right, the axes are different. So the one on the upper left is more distributed in order to accentuate the difference versus the one on the bottom. Is this completely incorrect or no? You're shaking your head. Why?
I think it depends on how significant statistically and clinically the difference is.
And to be consistent, if you're showing a battery of graphs, make sure that the axes are consistent. So your reader, whether it's a target audience in a talk or certainly in a publication, is able to make that comparison correctly. I'm going to show a couple of examples, not true case studies, but I want you to think about a type of approach when you're faced with a conflict or a case study, and that's called SFNO. Looking at the stakeholders, the facts, the norms, and the options. We never have all the facts to make a decision in a particular situation, or when we perhaps are reading a publication that we're not familiar with. Let's look at a couple examples and use this approach. So here's Eric Pullman from UVM, many years ago now, well over 10 years ago. He has the dubious distinction of being the first scientist to go to jail. It was found that he had fabricated data on his research with regard to hormonal supplements, had really garnered well over $500,000 in federal grant monies, but then someone blew the whistle. And it was found that he had committed misconduct and ended up going to prison for 1 year and a day. UVM had to pay all that money back, certainly plus the monies to conduct the investigation, which are not cheap. Guess what he's doing now?
Researching.
No, he's been barred from research here in the States anyway. He's a college counselor in Montreal. That hurts. He's teaching kids, man. Cody out of Duke, a little more recent. You may be well aware of this case. He had fabricated falsified data in his area of cancer research, and it was a biostatistician that blew the whistle. This too was a long, ongoing investigation. Duke had to pay back money. Cody was fired, barred from getting any more research dollars. He is now in Nebraska as a practicing physician. He's not doing research, but I'm not sure that I would want to have him on my team. UAB is not without incident. You may be well aware of this particular case. This was now several years ago as well, out of our Department of Surgery. There was a group that had been examining anti-rejection drugs and developing these novel drugs. And using a primate model, we're testing these drugs on primates that had received a kidney transplant. So in essence, they were taking these primates, removing both kidneys, transplanting one back into the primate, and then testing the anti-rejection drug. Fabulous results, near normal kidney function, primates were surviving, everything looked great. Someone blew the whistle. They, in fact, did not take out both kidneys, they took out one. So primates were surviving on their native kidney. 2 investigators were fired. Labs closed down, money had to give back. Well over $500,000 just to conduct the investigation. So in these series of examples, who are the stakeholders? Sorry? People. Be more specific. Who are our immediate stakeholders? Taxpayers. Come a little closer.
UABs.
So in this case, in particular, UAB. Who else?
People who would take the drugs, essentially.
Exactly. Patients, ultimately. Who else?
Members of their labs.
Members of their labs. Scattered to the winds. Trainees. Had to find new labs. Who else? Open it up a little bit more now. Funding agencies. Right? What if you're an investigator in one of these fields? You've read all their papers, designed experiments based on these bogus manuscripts. You're a stakeholder. Journals that published these papers, stakeholders. Excuse me. What are the facts? What do we know about each of these cases? Why do you think they acted in this way? What would drive somebody to falsify or fabricate data? Publish or perish. Pressure. Pressure to get a job. Pressure to graduate. Pressure to be promoted. Keep your lab going. Yep, lots of pressure.
What was the university's response back in 2009? What were the high-level discussions? What do you put in place to prevent it from happening?
Sure, and we're going to talk at the end about the approach to use if you do have a concern. This allegation was made to our Vice President of Research office. We have someone here called the Research Integrity Officer, or the RIO. And it's their job to conduct these investigations, to assess, initially, to assess the allegation, if it's credible, to move forward with a formal inquiry. At that time, there was a group, a committee convened of faculty in that area, so they well know how to assess the data, certainly know the field. I've been on one of these inquiry groups. It's gut-wrenching to have to investigate one of your colleagues in this way. If the inquiry group then feels, yes, there is smoke here and maybe fire, then it goes to a full investigatory committee, which is a different set of faculty. Lawyers become involved at that point. This particular case went on for 2 years in terms of an investigation. And ultimately they were found, as I mentioned, to be guilty, and they were fired. So it is quite a long process, but I think one that works, for sure. Other questions?
But you asked what the institution does to try to make this not happen. And this is one of the things that the institution does, is to try to raise awareness through discussions like this and as many venues as we can. NIH requires that this kind of discussion, that there be refresher training. Actually, I don't know what the period of time is now, every 3 years?
So it's full training every 4 years. It's preferred refresher every year. But yes, it has been identified as one of the key issues from the President's Risk Cabinet here at UAB, called the PRC. It is a cabinet made up of representatives from across campus that bring issues with regard to concern and potential risks that the university hopefully could avoid. And this was one of the 4 that were identified.
What, do people have ideas of other things that would help to
Good question.
create an environment where this didn't happen? I guess I can mention one other thing. There are anonymous hotlines where people can report things that they're concerned about so that they get investigated.
Yep, and we'll look at those points of contact toward the end. Maybe I can hand out a handout now, and I don't know that I have enough for everyone. And for those listening in, the handout that I'm using is, there you go, thanks, an infographic off of the NIH Office of Research Integrity website that you can go and peruse. It's the one on misconduct. No surprise there. But it does talk about the pressures at every level with regard to publication, getting grant money, certainly promotion jobs, and so forth. There are no easy answers. Our incentive model is built this way, and it's going to be hard to break. It's not to say that we have to do away with that incentive model altogether. I mean, I think it has served us well in the past, but perhaps we can attenuate or modify it in some way to decrease the pressure that people are certainly under and feeling. I raised this example, the MMR vaccine in autism, which again, you're probably well aware. This was the 98 paper out in the Lancet from Andrew Wakefield's group, demonstrating, air quotes, a link between the MMR vaccine and autism. Not sure how much you know of this particular study, but Wakefield essentially conducted his study by sampling DNA from cups collected at his 6-year-old son's birthday party from kids without their consent, went ahead, cherry-picked his data, and came out with this very short publication, The Lancet, linking MMR and autism. I'm hoping I have a internet feed, because if you don't know already, this was in this week's Scientist, measles outbreak in Washington State. There are, I believe, over 40 active cases right now, spreading like wildfire. This is taking off in a county, I believe, in southwestern Washington State, where only less than 80% of kindergartners have been vaccinated with the MMR vaccine. Less than 80%, which is well below the herd immunity percent of what, 92 or 95. There was a live infected person that went to a Portland, Oregon basketball game. Forget what they're called. So it's likely it's going to be spiking up now in Portland. But there are other, certainly, flashpoints across the U.S. where this has happened. And I know polio is on the rise as well. All because of this bogus study that has been retracted many times. You'll still see, though, in the literature, references to this particular study and others trying to replicate it. So it has well damaged that trust that we have. So we talked about stakeholders and facts, and certainly the norm is doing our research responsibly. And not much else to say there. UAB expands their definition of misconduct a little more broadly to include withholding information from an active investigation. Bringing malicious charges against someone can in and of itself be misconduct, as can any acts of interference or retaliation during an investigation. So UAB casts that a little more broadly to define. So we've talked about FFP and how that can dribble over into different other practices of misconduct. What's not misconduct?
Slightly adjusting image quality, sometimes it's too dark, so if you do it equally along all of them.
Consistently, okay. What else? We're human. What do humans do?
Make errors.
Make errors. Exactly right. But we also like to argue, which is always fun. Difference of opinion, difference of interpretation. That's not misconduct. I'll never forget, I went to my first really big international conference for cystic fibrosis. Demigods up on the stage in my mind, because I've read all their work. And they are yelling and screaming at each other about interpretation of data. I'm like, oh man, what about them? Really awkward and uncomfortable. but not misconduct. I certainly always wanted my trainees to be arguing with me, which meant that they had read the literature, that they had formed an opinion and an idea about what might be going on. But that's not misconduct. That's what you want. That's how you move the needle. So to bear that in mind, that there has to be intent in terms of the error, if you will. So as I mentioned at the outset, I want to really touch upon a little more deeply on two areas, data management and authorship. There are a number of different areas that funnel into data management. Ownership, certainly collection, storage, how to protect that, how long do you retain it, analyze, and so forth. So before we manage it, though, I think we should define it. What are data? What are they?
Outcome, the result. Yeah.
Outcomes and results. What else?
Any collected information.
Any collected information. Okay. There's more. Notes. Okay. How do you get these numbers? How do you get these notes? What leads you to that? Measurements. How do you take the measurements? Carefully. Good answer. When you start to do an experiment, what's the very first thing that you do? Sorry? Okay.
Warm up the media.
Warm up the media. Why do you do that?
You have a protocol.
You have a protocol. You have an experimental design. That's part of the data. The methods, the type of analysis that you choose, part of the data. How you calibrate the equipment if it requires that, or how you validate a questionnaire if that's a tool for your work. All of those pieces, the reagents, the tools that you use, are considered data, as are the samples, certainly, and the numbers that you get. So if someone were to make an allegation and the Rio comes into the lab, she's going to take all of the data, the samples, the hard drives, the notes, the presentations, understanding the experimental design, all of that feeds into the data. So all of those things then need to be managed, need to be stored carefully, and certainly critically written and carefully written, ultimately. Who owns the data? Is it UAB? Is it the funding agency? Is it the journal once it's published? Is it you? Who owns it? It depends. How does it depend?
Depends on the funding source.
It does. Okay. What do others think? Where does that funding source route through? OSP, UAB. Anything that routes through UAB to you, UAB owns it. So right, it could be NIH, it could be NSF, although NSF can be a little quirky if they sometimes will pay the investigator directly, a little different. But ultimately it's UAB. And certainly if it's any work done on UAB campus or space, it's UAB. If it's done by a UAB employee, it's UAB. So the institution does own it. How long do you have to retain the data? 5 years? Other guesses? 7 years. Anybody else? So here's where it does depend. And the funding agency sometimes, I believe it's 3 years for NIH after the funding has ended. Journals say 7 years after the last citation. So if you have a hot paper that gets cited a lot, you're going to have to keep that data forever, ultimately. And that can be a problem, essentially, especially for UAB. And think of our number of investigators that we have and just the volumes of data. So that they are struggling with how to back that up and keep that safe. What are the benefits of good data management?
Confidence in the results.
Confidence in the results, absolutely. What else? Say again?
Transparency.
Transparency, great word. Accurately analyze and assess the work. It's all out here. Accurately communicate the results. Rigor and reproducibility, we touched upon, we'll touch upon a little bit more. Certainly it details the rationale, provides justification to sponsors, and reliable and valid results. So if you have really strong confidence in the way your data is managed, you'll be able to do these things pretty easily. UAB is exploring purchasing a lab archives platform. Does anyone use an electronic lab notebook currently? We are going to be demoing or doing a pilot phase actually with several investigators here at UAB in a couple of months to see how well the lab archives platform works. And it's really applicable to basic clinical and translational science across the board. Fairly easy to use, as I understand. I've not used it myself, but we're hoping that if all goes well, we'll be able to purchase a license, and that would allow us to offer it to faculty and staff and trainees pretty cheaply going forward. Again, to help data management to avoid issues of misconduct. I think you'll be hearing more about rigor and reproducibility, certainly through the Kaizen game. But that is definitely a term now the NIH holds near and dear. I mentioned before the confidence from our congressperson. But definitely there have been several reports, probably well known, that you may be familiar in the news about inability to reproduce results. You're familiar with the Ambien story, the sleep aid? First tested and developed several years back now, forget the company, but they tested that in males. Saw that it was effective, efficient, safe, and so they rolled it out onto the market and began prescribing it to both men and women. Well, women began to sleepwalk, sleep drive, so forth. Why?
It ended up being like twice as potent in women.
Why? Why was it twice as potent?
Probably some liver enzyme thing.
Right, so it was a metabolic issue. Had the company initially tested this in women, who would have known that? Or men and women. So that's just one case and one example that has really driven the NIH to require now both sexes be included in any type of study that, say, it's mice or certainly clinical studies to avoid things of that nature. Excuse me. How many of you do cell culture in the labs? Do you use fetal calf serum? What's in the fetal calf serum?
No one knows.
No one knows. It's the secret sauce, right? When we would do cell culture, we would buy the entire lot because it worked, air quotes. But that is one example of how such a variable can differ from one lab to another, even on the same campus, and would allow for different results. So again, it's these types of studies, and I know you'll be diving more through the Kaizen game into this topic, but again, it is to make sure that we do our work responsibly. Certainly best practices, I touched upon this a little bit, careful record keeping, making sure it's systematic, that notebooks are organized, you follow your field's guidelines and standards, the norms for your field of data collection. I have embedded here, I think you're going to get the slides. There are a few links that you may find fun that are videos that we don't have time to go into today. But the electronic records one that is highlighted is linked to a case study on the ORI website about storing making sure your electronic data are backed up. And in this particular video, the computer crashes and the postdoc loses the raw data. He goes running to the mentor saying, I have a graph of summary data. We can use this. Can they? They have a grant coming up, a grant deadline in 2 days. They really needed the data. They don't have time to repeat it.
They don't have like a lab notebook.
It was all electronic. It was supposed to be backed up.
I would say it's not gone.
It's not gone. So you have a summary graph. Can you use that?
The data's not gone.
The raw data are gone.
Assuming like the computer didn't catch fire or anything.
They can't get it. Assume they can't get it, and that's what they do in this video. What do others think?
I think it depends on what you're using it for.
It depends.
I think there's a difference between saying, look, we saw this in a grant, we saw this before, we intend to repeat it, and then repeat it, make sure you have the data. If you're publishing on something that you can't access the raw data, that might be a little bit more.
I would caution that if you don't have the raw data, don't go there. If you are ever called on the carpet, and even funding agencies may ask for raw data, and you don't have it, it's not pretty. So summary data aren't enough. You need the raw data. But go and look at these links. They're pretty fun. So a lot of what we do now is team science. Big groups tackling big ideas, whether they're here at one institution or across the country or global. And how do you manage that team? Again, because you want to make sure that things are consistent and responsible. Definitely set basic ground rules of data management, and you're probably familiar with this if you're involved in any pharma study that have multiple sites. You have to have a consistent protocol across all of those sites of how the data are collected and analyzed. Definitely to create a flow chart of responsibilities and communicate. Be consistent, communicate often to make sure that everyone is on the same page. And this is particularly important with regard to authorship, any IP that may come out of the study. Certainly having team meetings is important. But authorship in particular can be sticky. We do have finally an authorship policy here at UAB. It's not that old. We've only had it less than 5 years. Shocking. What is in that? I can't talk. What is in the policy? Do you know? Are you familiar? So it's based on the ICJME International Committee of Medical and Journal Editors. It's probably not surprising, but we expect that those who are listed as authors really contributed to the conception, conduct, and design of the study, as well as the analysis and interpretation of the data. And those who didn't should be listed in the acknowledgements. So no honorary authors. Do you know what an honorary author is? Maybe putting your cat on the publication. You laugh. It has happened. When I was a postdoc 100 years ago at Hopkins, I was in the clinical immunology division. And at that time, the director was pretty strong of having his name put on every paper that came out of the division. Well, whether he knew you or not. And that was the norm at the time.
Like last author?
Just buried in the middle. In the middle. So if there are any authorship disputes, those are really taken to the level of the chair. Those don't go up to the RIO, the integrity officer. Those are really to be chewed on first at the chair level. And then if there are still concerns, there are other ways to expand that conversation. Have you heard of predatory journals? I definitely want to make sure that you knew of these. Has anybody received an email to be on the editorial board? You have. You're shaking your head. Yep. Don't agree to that. So these are research journals that have really popped up, I'd say, within the last 5 to 7 years, maybe. They charge incredible rates for publication, but there's no peer review. They just send it. They just publish. They notify you of the charges only after it's accepted, and it's accepted like the same day. They target scholars through mass emails to participate on their editorial boards or maybe present at these meetings. You can be a plenary speaker, invite 10 of your friends, and all come. It's a Ponzi scheme. Those particular meetings are Ponzi schemes. Don't list them on your CV. They're not real invites. I know people who do. They're not real. Editorial boards I mentioned, fake scholars. Cool. And they're trying to get credibility, right, for their journal. They'll actually take the design and branding of real journals to make themselves look real. They use impact factors as well as ISS centers. So again, you're going to get these with the links. I just want to show you the list. That's just A. That's B. You can keep on going, and you can pick out different ones. Some look real. Some look really real. And there's a lot of dispute. I know of or have read that some of these journals have filed lawsuits against this particular person that initially spearheaded the predatory journal investigation, maintaining that they're real and so forth. But just a word of caution. If you do receive one of these emails, I found myself on some immunology board, couldn't get myself off, finally gave up. Just buyer beware. Make sure if you do get an email or see one of these journals, and some of these will actually come up on PubMed. So be careful. How common is scientific misconduct? I've included some links here to show kind of an increase over time, as well as the list on the ORI case summaries. And yes, that UAB example that I gave is listed there in 2009, as we've had a few others more recently as well. But here's some of the data that's also posted on ORI. And this is a graph from 2006 to 2015 showing the number of reported inquiries and investigations conducted. So of those investigations conducted, I think you can see that really a fraction really found misconduct. And of those that were deemed to be true misconduct of these allegations, most fell in the buckets of the FFP, right? Falsification, fabrication, largely. So who done it? Was it the grad student? Was it the postdoc? Was it the faculty member or principal investigator? Who was the primary person for these reports that was responsible for the misconduct? Trainees? So I have a trainee guess.
Typically the guy on the bottom.
Typically the guy on the bottom. Any other guesses? Okay, roll it out. So difficult to see, so let me help. So these first three bars are essentially the trainees. Student, tech or research assistant, postdoc. Postdoc, up to 30. But here are mentors, if you will, assistant, associate, and professor. Add up these three bars. What do you get?
35.
It's a little bit more than the postdoc, right? Again, pressures.
I've seen a lot of assistants almost work like postdocs.
I'm sorry, can you say that again?
I said I just see a lot of assistant professors almost work like postdocs.
So the comment is a lot of assistant professors work like postdocs. Yeah, you're establishing yourself, you're in the lab, writing the grants, you're not sleeping. Yeah. Interchangeable, sure, but the pressures are different. For postdocs, it's getting the publications out, maybe getting that K award to find a job, to get the job. And then once you get the job, you want to be promoted and tenured, right? And keep the lab going. It's pressures. And there are no easy answers of changing this incentive model. But I think we need to be critically aware, centrally and then certainly departmentally, as to how to hopefully attenuate that pressure. If you are under pressure, reach out. Certainly anyone in administration, CCTS, grad school, are always happy to help and help you think through or talk through some concerns. If you do have a true concern that you think needs to be reported, there is a tree that you can kind of hierarchy-wise go to. But as David said earlier, you probably want to do this anonymously. There is an ethics matters a hotline or website that you can report your allegation through. And this routes through a third party. So you don't have to give your name. In fact, they don't want your name. They'll give you a case number. So that if you ever want to call back and check on the status, you have to give the case number, not your name. That allegation routes to this person, Pam Bunellis. She is our research integrity officer and the one in charge of assessing these initial allegations, forming an inquiry team if needed, and ultimately conducting the investigation. Before I get her contact, just one final note. We lean on Uncle Albert. Many people say that it's the intellect which makes a great scientist. They are wrong. It's character. So here's our additional informational resources. The handout that I passed around is on the ORI website. I failed to mention the Authenticate plagiarism software. UAB has now secured a contract with Authenticate. If you are familiar with Turnitin, it's Turnitin on steroids. It's essentially the same group, a larger database that they compare against. And this is now accessible, as my understanding, free to faculty, staff, and trainees now at UAB, just off the press. I mentioned Lab Archives, that's in progress. Rigor and reproducibility, I know you hear about for the Kaizen game. And contact, and certainly there's Pam Pinellas' contact directly if you have any questions. Questions for me now? All right.
One of the most difficult issues I've encountered is the evolving concept of self-plagiarism. And it comes up in a lot of different ways. So one is that there are most journals have very clear guidelines about what's acceptable in terms of showing in a publication that you're submitting data that's already been published and how you should handle that. And I don't know, maybe Lisa, you'd want to make some comments about that.
Right, so what are you most likely to self-plagiarize? When do you think?
When you're writing more than one manuscript. When you're writing more than one manuscript on the study you're doing, maybe a protocol manuscript, maybe.
It's the protocol, it's the methods section. Once you have submitted that for publication and it's published, it's owned by the journal. So going forward, if you still use that same type of experimental design, you have to reference that. But to the question of data, can you ever use the same data in different publications? It depends. If it's a data set that you're using to chew on a different question, that's allowed. But you have to make that crystal clear. I think that's what you're referring to.
Right. I mean, for sure, you can't say, I did the control experiment 3 years ago, and I'm not doing it again with this current experiment because I already did that. That's not acceptable. But I guess another area where it comes up, and if you are old enough like me to have written a review kind of article, and then you get asked to write another review on the similar or the same topic, or just to update a chapter in a book, how you really handle that. I mean, you can't write the whole chapter over again completely. So you just need to really work with the publisher to figure out what their guidelines are. But it's a
very true. I mean, and it's difficult, right? There's how many different ways can you say the frog is green? Sometimes there are accepted statements or accepted knowledge that you don't have to reference and that you can use. It really depends on the target audience for the journal in particular times. But right, to make sure you don't fall into the trap of certainly typing and writing what you just read. I know I sometimes do that, and I have to catch myself. So. Other questions? Yes?
What are the, sorry, this is just for UAB.
Sure.
What are the, trainings that we should be doing every year? Sorry, I just thought that we were supposed to be doing them every 3 years.
Well, it's full training every 4 years. It's recommended that you have a refresher like this once a year. There are modules, as I mentioned, online that you can tap into. They're called City, and I can provide instructions on how to access that. Or we always have a number of workshops that are ongoing. If you want to do a deep dive, you can come visit me for 8 hours on a day, like tomorrow. I do 8 hours of stand-up or CR all the time. But that's probably a too deeper dive for a refresher. But there are a number of workshops that I conduct across campus like this for that tune-up.
What is the every 4 years?
Every 4 years is the formal deep dive training. And we are now establishing requirements for faculty and staff that do touch research in a heavy way, certainly if they're federally funded. And that's going to include really a set of modules, the online City modules.
One of the things that's recommended is if you're a lab leader and you have your own group is to dedicate a lab meeting once a year to RCR update and to have, I mean, I think Elisa's office or the Office of Research Integrity website has little modules that can be stuff you would discuss with your own group once a year.
There's some really great resources on the ORI website.
Right.
Really great.
Right.
And they're short. That's fine.
Yeah, there's actually a really nice set of videos where you can take the part of a different person and see how a scenario plays out if you're looking at it in different ways. And
it's entertaining. You can make them
choices along
choose
the way.
badly and get them fired. It's
Right.
kind of, students like that.
Sort of fun. Okay, well, thanks very
Thank
much.
you.
Thanks a lot, Lisa. So we'll take a 5-minute break, and the second part of today's session is going to be an introduction to research ethics as it particularly relates to genetic types of research, and I'll introduce that when we come back. So take 3 or 4 minutes to stretch your legs.
Hey there, how are you? You've got some good examples to chew on. So, yeah. Let's read more about the CRISPR. Yes. Wow. Yeah.
Have you worked with this system before?
No, not really. And I heard of pump drive.
Okay.
It's possible to opt.
Yeah. Yeah, you can put it in. And well, that's not bad.
Sorry, I can't stay. Take care. Thank you.
One thing you can't appreciate. They do it. And then they're like,
Okay.
I like how they were all like performing.
Sure, possible. Right. Actually, it's the person who runs our tech that is coming out. So we have quite a few people joining online. So we have to use the assessment here. So you can change the slide with the arrows for. I mean, this is a that shows up in his season also. So it darkens the whole screen.
Okay. I sometimes think that very good.
And then I will work on it here to try to have questions or comments. It doesn't sound like it's working here, it's just showing online. But if I don't get to somebody, if they speak out too fast, if we can repeat their question or somehow interpret it so that the online people know the question.
Okay, yeah, that's actually good.
Okay, I think we'll get started. Did anybody not get a handout? I have a couple more here if you need one. Yeah. Okay, so the second part of the presentation today is going to be delivered by Dr. Mariko Nakano, who's in the Department of Medical Education here at UAB. Dr. Nakano has a PhD in ethics from Kyoto University. She worked at the Ohio State University before she came to UAB about 4 years ago.
Yes.
And she's now the co-lead of the ethics component of the Alabama Genomic Health Initiative, which I don't know if she'll describe specifically in this talk, but it's a terrific effort to try to use prospective genetic testing to see whether that has an impact on the long-term health of the populations in which it's implemented. There are a lot of ethical issues that come up related to genomic research in large populations, particularly very diverse populations. And so I encourage you to engage in dialogue over this. There's a lot of things that are well worth talking through. Thanks for being here, Nick.
Thank you, Dr. Jackson. So I'm Mariko Nakano from the UAB School of Medicine, Department of Medical Education, and I'm a PhD in ethics and bioethics. So today I'm going to talk about bioethical issues and population-based genomic and genetic studies. And I want to discuss this from my experiences of the AGHI, the Alabama Genomic Health Initiative, which is a statewide genotyping / genome sequencing study, which started in 2017. So it's funded by the state of Alabama, and it's a joint project between UAB and Hudson Alpha Institute for Biotechnology. And PIs are Dr. Bruce Cove at UAB and Dr. Greg Arch at Hudson Alpha Institute for Biotechnology. And I'm serving as co-chair of this bioethics working group with Dr. Tom May from Hudson Alpha and Dr. Stephen Shodecki from Tuskegee University National Bioethics Institute. So now I'm not a geneticist, so excuse me if I use some scientific terms incorrectly, but I will hopefully be able to provide valuable insights about the ethical issues of this type of research. So this is a speaker disclosure and disclaimer, which you can find on your handouts. So let's see what's going on in the world of genomics. I assume that we all know 23andMe, right? And this sort of genetic testing services became possible because of researchers' huge efforts to find correlations between genes and health conditions through population-based genomic research projects. Prior to the rise of the DTC companies, direct-to-consumer companies, there were various biobanking projects from the late 19th, such as Icelandic tree database project in late 1990s, and Estonian Genome Project, and UK Biobank, and similar biobanks in China, Mexico, Canada, Japan, and others. And today, we have even more advanced biobank and DNA database project. For example, All of Us project aims to collect genetic and health data from 1 million subjects. And outside of the United States, FinGen study in Finland is a unique study that combines genome information with digital healthcare data through the nationwide collaboration among Finnish universities, hospitals, and biobanks. And the samples will be gathered by nearly all biobanks in Finland, and it aims to collect samples from 500,000 participants by 2023. And at UAB, we have the statewide population genomics project called the Alabama Genomic Health Initiative, or the AGHI. And enrollment is currently done in Birmingham, mainly in Brookline Clinic and Medical Tower, and also in Huntsville, Selma, and Montgomery. And some pop-up enrollment events are also taking place in various rural communities. And the research team plans to recruit 2,000 participants per year with the goal of enrolling 10,000 participants over a total of 5 years. And as of January 25, 2019, just last week, over 4,100 people have participated in this study. And this is the basic structure of the AGHI. We recruit 2 cohorts, one being healthy individuals and the other being affected individuals. And healthy individuals mean people who are either healthy or are obtaining care for various types of health problems with no specific target for this study. And affected individuals, it's hard to use it, so I won't be. Affected individuals means people with clinical signs and symptoms that suggest an undiagnosed genetic disorder. And this cohort may contain very young individuals and children. And both cohorts provide a blood sample. And the samples from healthy volunteers will then undergo genetic testing and be analyzed for variants that are known to cause significant health problems for which there are proven effective approaches to treat or prevent the disease or improve the outcomes. So we call it medically actionable results. And as of now, we use ACMG59, which is the list of variants in 59 genes recommended as medically actionable by the American College of Medical Genetics and Genomics. So the ACMG59 results will be returned to participants. And in case any medically actionable item were found, the participants will receive medical advice from genetic counselors. And these ACMG59 results include variants related to tumor predispositions, such as BRCA1 and 2, or MLH1 and others, and connective tissue dysplasia, and cardiac diseases, or metabolic disorders, or pharmacogenomic conditions such as malignant hypothermia, which is a type of severe reaction that occurs in particular medications used during genomic anesthesia. And on the other hand, the affected cohort undergo whole genome sequencing to identify a genetic variant that explains the conditions. So diagnostic results will be returned to the participants and genetic counseling will occur. But this is not the end of the story. In addition to this genotyping and sequencing, all participants will be requested and asked to allow storage of biospecimens, blood samples, and to answer a short medical questionnaire, and to permit medical record data to be stored in the study databases such as I2B2 and ENCORE. So genomic data obtained by genotyping or sequencing will also be stored in ENCORE. And these data will then be used for research on genomic contribution to health and diseases. And made available to outside investigators with IRB-approved studies. And these outside studies will also be subject to the internal review of the AGHI steering committee. So this project has both the clinical and the research aspect. This upper part has clinical importance for participants, whereas this lower part is purely about research that will hopefully advance knowledge and benefit future patients. So it's a complicated study that requires lots of collaboration. So the AGHI team consists of multiple working groups, so including recruitment and community engagement working group, and bioinformatics working group, and genomics working group, and bioethics working group, among others. So working group leaders update each other on a weekly basis and meet and discuss important issues on a monthly basis. Then what sort of ethical issues should we anticipate? Ethical issues of genetic testing, screening, whole exome sequencing, whole genome sequencing were extensively discussed by the Presidential Bioethics Commission several years ago. And a very thick report entitled Privacy and Progress in Whole Genome Sequencing was issued in 2012, which discussed the issue of stigmatizing, privacy issues, and familial implications of whole genome sequencing. And its message was rather simple. It said, progress is important, but privacy is also important. So de-identification of the data and informed consent were the two main norms they proposed. But we have actually encountered many specific issues at each step of our project, so I will explain each of them later. And ethics of genomics and genetic research is important in today's context, and especially in Alabama. First of all, there remains distrust in research here in Alabama. If you remember the notorious Tuskegee civil study, there's no wonder there are still people who don't trust scientists at all. In addition to that, recent trend in bioethics should also be considered. Nowadays, we conduct more and more translational research, and that has been clinical, that has both clinical and research components, just like the AGHI. And this could cause confusions on the part of research participants who are also patients. They may be misled to believe that the research project they sign up will bring about huge therapeutic benefits to them when it really doesn't. And this is called therapeutic misconception, and researchers have ethical obligation to avoid that. Also, in research using database and biobank, there's greater concerns about how to protect participant privacy. We should also keep in mind that once somebody's genomic information itself is personal information. As we will see later, now, the AGHI research team and bioethics working group team regard each participant's genomic information as personally identifiable information, and we don't call it anonymous or de-identified information. Also to be noted is the current trend about the ethical rule of fairness. Previously, the Tuskegee civil study, in which only male African Americans were recruited for the study of syphilis, and the Willowbrook hepatitis study, in which mentally disabled pediatric inmates were used to study hepatitis. And those studies were accused of unfair inclusion of vulnerable population. But nowadays, unfair exclusion of vulnerable population from the study is also considered unethical, because those populations may fail to receive benefits of medical advances by being constantly excluded from research studies. So we have regulation, and so we do have the shifted focus about fairness, and nowadays, unfair inclusion as well as unfair exclusion from the study is considered unethical. And also, ethics of cutting-edge biotechnology may not be fully covered by existing policy and regulations. Now, we do have regulations at the federal level, such as HIPAA and GINA. HIPAA states that it is illegal to disclose a patient's personal health information to anyone outside of the healthcare team that provides care for the patient. The GINA prohibits Genetic Information Non-Discrimination Act of 2008. GINA prohibits discrimination in health insurance coverage and employment based on genetic information. So these are two ways to ensure patient privacy and confidentiality. But we know these regulations won't give full protection for research participants. Genomic information itself is personal information, as I said before, and I will explain more fully later. And we should also be aware that GINA does not cover discrimination in settings other than health insurance or employment. For example, situation in education, discrimination in marriage, and others. And GINA also does not protect participants from possible discriminations in life insurance, disability insurance, or long-term care insurance, or employment at private companies with fewer than 15 employees. So there are some loopholes in GINA as well, current existing regulations as well.
Can I ask you a question on the first bullet point on the last slide? Do you have any data on the proportion of people who cite distrust in research or how that's changed over time? In my HIV clinic, I hear this distrust every day.
Yeah, concrete data, but this is what we feel. And we try to figure out, get some data from investigating into who declines to participate in the AGHI, for what reasons, but it's hard to obtain that data. Any other questions? Okay. So having these recent local trends in mind, the AGHI Bioethics Working Group has proposed 4 basic values that should be held throughout the research project. These are transparency/honesty, and respect, confidentiality, and fairness. We didn't come up with these 4 at the beginning, but we gradually came up to form these 4 core values as we went through lots of discussions. And we will come back to these 4 basics later, but let's focus on more specific ethical issues at each step of this sort of large-scale population genomic genetic studies. So let's see what our researchers expected to actually do. I will briefly overview the ethical issues of research and clinical aspect of the AGHI. In the research aspect of this sort of research, we encounter issues of broad consent, protection of patient privacy, and how to return results to research participants. In the clinical aspect, we encounter the ethical issue of avoiding therapeutic misconception in patient participants and of how to respect potential participants. So first, AGHI has had to tackle with some bioethical research ethics issues at each of 3 steps: data and sample collection, and storage, and data mining. So let's look at ethical issues about sample collection. Informed consent process starts with recruiting. In the Tuskegee syphilis study, participants were recruited by being told that all Black men had bad blood, which was inaccurate, deceptive, and unethical. And so we wanted to do the right thing from the very beginning till the end of this community engaging project. So we needed to carefully define who should be recruited, whether we want to include minors, incompetent patients, and other vulnerable people for whom we need to devise extra protection and precaution. And we needed to carefully define what to explain to such potential participants when recruiting them. And we thought we must disclose conflict of interest while carefully explaining how important this project is for current and future patients and for the general health of people in Alabama. And after recruiting potential participants, we officially obtained informed consent. And one ethical issue at this step is whether it is okay to obtain broad consent to our project. Now, specific consent means research participants give consent to a particular research project that has a specific aim and specific duration and an endpoint, like a typical clinical trial to test a certain experimental drug. As regards to large-scale population genomic studies, such as AGHI, however, we want to utilize a database and biobank for unspecified broad research purposes for unspecified period of time. So we needed to obtain from participants the consent for such broad uses of their samples and their health data. So we had to discuss whether and how we can ethically justify the use of broad consent. We also discussed what to inform potential participants and ways to inform them. So we discussed ethics of broad consent and consent for minors, and what to inform potential participants and ways to inform. And after samples or data are collected, they are stored in a database or a biobank, which comes with the ethical issue of security and privacy. So we needed to get prepared for possible data hacking or inadvertent data leakage. So we also had to clearly define who should access the database and who should manage the identification key that links identifiable personal information and de-identified data. We also needed to delineate contract-based obligations of data managers or honest brokers as regards to confidentiality, safety, and the veracity of data. When the data manager leaves the job, we make sure that he or she continues to keep essential information confidential. And there are also problems that could occur from sharing data statewide or nationwide. The biggest issue is possible re-identification of sample donors. I'll explain this a bit more in the next 2 slides. And there's also the question of what to do with novel clinical findings or secondary findings we obtain through research, and whether and how we should give any feedback to participants or biospecimen donors. Now let me explain the issue of re-identification. Even though the database and biobank are supposed to be carefully de-identified, stripped off of all HIPAA-designated 18 identifiers, such as names and street address. Even though with all that, whole genome sequence data are unique to only one person, making them vulnerable to re-identification. For example, in 2013 science paper, Melissa Jimrak and her colleagues at the Whitehead Institute at MIT reported that they could successfully profile the full identities of 50 individuals who participated in public sequencing projects. What they did was this. The researchers first obtained the de-identified sequencing data sets from public sequencing projects, which are just like ours. And then they profiled short tandem repeats on the Y chromosome, so YSTRs, of those anonymized participants. And they use the data on Y chromosome because it's unique to each biological family inherited from father to son. Then they cross-references such YSTR data with recreational genetic genealogy databases such as Ancestry.com or Family Finder. Then they could infer surnames of not all, but many of those de-identified genetic data. Then the researchers searched those surnames together with other metadata, such as age and state, using people finders or white pages, which are also free and publicly accessible online databases. And as a result, researchers could figure out the full identity, including full name, address, and phone number of multiple targets. Not all, but multiple targets. So we need to be ready to handle such re-identification of sample donors. And more recently, as some of you may remember, in the early summer of last year, 2018, a DNA website was used to identify the Golden State Killer, Joseph D'Angelo, who is said to have killed 12 people and raped 45 women across California between 1976 and 1986. In this case, the California investigators used the biological sample obtained from the crime scene and extracted DNA data from that sample and uploaded this DNA data onto JADMatch, which is a DNA and genealogy research database, which you can see at the center. Then the investigators could find this DNA sample owner's distant relatives, which was actually third cousin and fourth cousin. And using this relative information, the detectives screened the family trees using the suspect's inferred age and the probable place of residence. Probably he should be living in California. And then they narrowed down the list of possible suspects to one single individual. And actually, US police now uses similar tactics to crack more than 19 cold cases during last year, which led to the arrest of suspect murderers and rapists. And interestingly, the success rates of this tactic are higher than with surname inference from the Y chromosome, according to a science paper published last November. And the same research team project that about 60% of the searches for individuals of European descent will result in a third cousin or closer match, which theoretically allows the identification using demographic identifiers. So the researchers say that the technique could implicate nearly any US individual of European descent in the near future. So there's issues about the possibly re-identification using the genetic data. So if population genomic researchers post their research participants' genomic data in a publicly available database or somewhere, some database which is publicly accessible, it can be easily used to identify that participant as well as the participant's relatives. So this is a huge threat to research participant confidentiality and a threat to the trust and trustworthiness of a genomic research project. Then there are ethical issues with data mining through whole genome sequencing or targeted resequencing. The biggest issue is what to do with incidental findings or secondary findings. Researchers may find a person's genetic predisposition to a disease treatable with early intervention, such as familial adenomyomatosis, or they may also find a mutation leading to a currently incurable disease, such as Huntington's. Or the sequencing may incidentally reveal the fact that a sample donor is not a biological child of his or her parent. So we have faced the questions of whether to inform sample donors of such findings and whether we should include a pre-arrangement clause in the informed consent document to let participants know how we handle such incidental findings. And this slide shows the AGHI's our attempt to handle all those ethical issues. Now, we decided to obtain broad consent from potential participants, but we tried to convey everything we know and everything we do not know about future users of participant data and biospecimens as honestly as possible. And in the informed consent document, we clearly stated that we will use participant sample and data for unspecified future research, but at the same time, we promise that those will be all medical research individually approved by the IRB. And also, we gave the choices for the potential participants whether they want to have their data and biospecimens stored in the biobanking database, and they can make choices. Also, to better protect participant confidentiality, we also obtained the Certificate of Confidentiality from the NIH. Now, the certificates of confidentiality are the formal certificates which all allow the researcher to refuse to disclose identifying information on research participants in any civil, criminal, administrative, legislative, or other proceedings, whether at the federal, state, or local level under the participant, unless the participant consents. And as for returning results, we included in our informed consent document a clear statement to return medically actionable results only, which means results about ACMG 59 at present, although we also stated that this list may be expanded in the future. Meanwhile, we also mentioned that we may possibly recontact former participants regarding new findings. And to be honest, we cannot guarantee that we will be able to return all new findings to all 10,000 plus participants in the future, which will depend on our technology and budget available to us each year. And we think we should not make false promise. So we stated that we will surely return medically actionable results, but refrain from stating we will definitely return all results that will become available in the future. But we will at least do our best to disseminate novel findings through our websites, public relations, etc. And we may possibly try to recontact to convey new medically significant findings as long as our budget allows. Now let's turn to the clinical aspects of the AGHI. Some sample donors are healthy at the time of sample donation, and others are patients with cancer or other devastating diseases, and still others are people with a family history of such diseases. And we needed to consider how to inform and counsel and heal such patients. And now informing. When we explain our project to desperate patients, we need to avoid therapeutic misconception. Now, patients may regard the blood draw as part of treatment, and they may believe that they are receiving some kind of medical care by providing blood to AGHI. And we should avoid such misunderstanding. And counseling. When we counsel patients for the presence of certain medically actionable variants, we need to consider how to explain such findings to patients. And also, when we find nothing that are on the ACMG list of disease-related genes, we may tell participants that we have found nothing. But then the participants may mistakenly believe that they are perfectly healthy now and won't have any disease in the future. So we need to educate people that genotyping or sequencing may not be a perfect predictive tool for healthy individuals. And finally, providing personalized care. When we provide personalized care for patients, we need to determine what health condition to treat and to what extent. For example, do we endorse preventive organ removal for people with BRCA1 or 2 mutations? So those are the questions we encountered. So these are the kinds of ethical issues that pertain to the population-based genomic/genetic studies that have certain clinical components like the AGHI. And this is a policy we developed to promote ethical interactions with patient participants. In addition to developing informed consent forms, we had training sessions with patient navigators and instructed them to emphasize the following. One, we try our best to clearly explain the research nature of the initiative. While we strive to offer medical benefits to each participant as much as we can at this point, the ultimate goal of the AGHI is to promote our knowledge about genomic medicine through biobank and database research and to benefit patients in the future for the generations to come. And we also try to honestly explain that the direct benefit to our participants may be limited. And those who will receive positive genotyping results will be 1 to 3%. Which means that more than 95% of people in the population cohort will receive negative results. And they may be misled to believe that they don't have to screen, do the mammogram or other regular checkup or something. So that's wrong. So we need to avoid such therapeutic misconception. And we also disclose research risks, physical risks, privacy risks, and group risks. And then we cautiously explain the meaning of results to avoid misunderstanding. For example, both patient navigators and genetic counselors are instructed to emphasize the following things. For one thing, patients shouldn't be too relieved upon receiving negative results. They need to explain that some people develop breast cancer without having BRCA1 or 2 mutations, for example. And participants should continue following medical advice and continue to do the routine disease screening. And also, the AGHI uses SEMG59 gene list initially, though we are discussing to expand the list. And other, on the other hand, other studies report different medically actionable gene lists. For example, NHLBI Grand Opportunity Exome Sequencing Project uses 112 medically actionable gene lists. And Gelsinger Health System and the Regeneration Genetic Center study uses 75 gene lists. And our current participants will receive SEMG59 results only. And they should be informed about that. And also, we decided to give potential participants some choices. For example, on the informed consent form, the participants can choose whether or not they want their specimens and specimen-related data to be stored in the biobank and used for future research. And so they can say, I agree to allow my specimens and specimen-associated data to be stored, or they can choose, I do not agree to allow my specimens and data stored. And as of last week, consent to biobank on this consent form is 91%, which is pretty good. And the AGHI participants can also choose whether they want to share the genotyping results with their healthcare providers. So they have three options. I would like to know results about medically actionable genes, and I want my medical provider to know results about medically actionable genes. And B, I would like to know results, but I do not want my medical provider to know results. And number C, I do not want to, and I do not know results myself, and I do not want my medical provider to know, which is that we added this option just for the sake of fairness. And as of January 25, 2019, as of last week, 41% of participants have consented to share results with the provider. And others chose to receive the results by themselves alone. But it depends on the situation. Once they receive the medically actionable results, positive results about the gene, disease-causing gene, they may often change their mind and change their choice on the informed consent form and ask us to tell the provider so that they can be prepared. And one more thing we greatly care about is how to strike the balance between accuracy and clarity. Because we are trying to convey complicated messages, our informed consent is 13 pages long, and it contains scientific terms which may not be easily understood by potential participants. At the same time, if we draft the consent form all at the 8th grade level, reading level, the information we want to convey may lose accuracy and may be misunderstood by participants. And for this reason, we have patient navigators. This is the patient. This is a patient navigator team. And for this reason, we have patient navigators who digest our messages and help assist participants by going through the entire informed consent process together with each patient participant. And if they hear some concerns from participants, they contact us and they question us. They ask questions to us and how to handle a situation like this. And as part of our policy on ethical interaction with participants, we also emphasize respecting the will of participants. So if a person expresses concerns and refuses to participate in the AGHI, we, the bioethics team, have a bioethics training with the patient navigators and genetic counselors and site coordinators. And we emphasize, we provide bioethics training and emphasize this. Don't coerce, don't persuade him or her by misleading explanation. And instead ask him or her to fill out the non-participation survey if possible. And if a participant is upset or expresses concern about the involvement in the AGHI, they are supposed to immediately notify the bioethics working group via email or we can use a call. And the bioethics working group will closely collaborate with other teams to better address all possible ethical concerns. And I think we have a great teamwork. And if there's a concerns of the confidentiality or data security, we immediately discuss the matter not only within the bioethics team, but also the bioinformatics teams and the biobanking team. And if there's a concerns about the returning of results, we discuss the matter with not only within the bioethics team, but also with the PI and also genetic counselors team to help solve the problem. So getting back to our 4 core values, we believe that these will be a helpful basis of ethical operationalizing of population-based genomic and genetic studies. So under the item of transparency and honesty, we want to mean that we should be honest about research benefits and risks, and we should be honest in explaining things to potential participants, and we should not deceive or mislead the participants. And under the item of respect, we try to honor a patient's choice to participate or not to participate, and we try to avoid exploitation or stigmatization, and we try to respect privacy as much as possible. And as for confidentiality, we keep sensitive personal information confidential. we don't say that we can keep sensitive personal information anonymous or de-identified, because it's not true. We regard a person's genomic information itself as identifiable information. But we can do our best to keep those information confidential. And as for fairness, we try to reach out to diverse population, and we think we should not exclude or exploit any specific group of people. So those are the what we want to mean by these 4 core values: transparency, honesty, and respect, confidentiality, and fairness. Now, to be honest, our ethics is work in progress, and there may be better ways to resolve those ethical concerns, and we may be mistaken in not addressing certain important ethical issues. So we try to be open to any questions and potential concerns and criticism, and simply try to do our best in behaving the most ethical way we can possibly conceive of. Anyway, what I shared with you today was a brief summary of the ethical issues we have encountered so far and how we are trying to handle those ethical issues. Okay, I hope, yes, so my presentation, so questions.
Are results returned to patients with a genetic counselor? Or, I mean, how do they?
As for the negative results, we send a letter to participants, and if we find positive results, the genetic counselor will call and try to reach out.
I have another question. There is, I think, a case in the UK of a daughter of a man with Huntington disease who is suing the physicians for not disclosing to her that her father had Huntington's disease, and then she was subsequently diagnosed, and by that time she had had a child. And her argument was that her decision about whether or not to have a child would have changed. And I think she won the right to sue the physicians in the UK. And so I think this definition of who we're obligated to tell things to may be a little bit in flux. So how's that being looked at in the?
Well, in the United States, the Huntington disease is a very touchy topic, and so we think that anyone who reached the age of adulthood should have the right to decide whether they want to be informed or not.
Right, I understand that, but I think the question is I think there's a discussion that's being had as to our obligation, you know, as physicians and providers on who we have to disclose results to. So not necessarily testing children, but and not necessarily just specifically for Huntington's, but this particular woman's argument was that her decision regarding her reproductive life would have been different. And so I don't know what the discussion would be.
Okay, as for the AGHI, we clearly stated that we will not return the results of the Huntington's because it's not on the ACMG 59 list. And if the potential participants want to know about their genetic status of the Huntington's disease gene, they should go to clinical genomics.
Removing Huntington's from the picture, that's just that particular case. I mean, genetic information by definition has implications for family members. And so, you know, there are BRCA1 change, if you find that, and that is on the ACMG list. If you find that has implications for potentially that patient's, all of their female family members in terms of screening and things like that. And I know right now in the US, you give that information to the patient, but not necessarily their family members. But, you know, I think there's a discussion about what is our obligation to families.
We explain the family implications of the, the genetics results. So we explain that you're encouraged to discuss with your family members, or with your family members. But we do not, decide whether they should receive the information. That's what the family should discuss. Okay.
Yeah, this seems really complicated to me because the researcher or the physician has the relationship with the patient, and doesn't have a relationship with the other family members necessarily. If they all came together, that would be different. But if
This UK case is she's suing the hospital. I mean,
yeah, that's
and it's really frightening as a provider that, or as a physician that that's a lot.
for sure. And you know,
Yeah.
how far into the family structure do you have to go to give fair information? It seems very like a very thorny, difficult path to navigate.
Population genomics studies like this, if that, you know, if she succeeds in that, I mean, that's the UK, right?
And but if she succeeds in that case, I mean, what's stopping people in the U.S. from bringing cases like that? And that's really frightening.
The research project, I think it's very important to clarify that what information we can provide, what information you need to go to the clinical genetic testing.
Yeah.
I think the question is, do we need to include something about like untreatable disease? So if somebody get my blood sample and I would like to know if there are any genetic diseases that they have, even if they're untreatable, like Huntington, that could happen to me in the future. In this consent form that you presented, there is no option of disclosing the untreatable diseases that I have. But do I have the right to tell you, hey, I want to know like all of my genetic profile, even if it's something that's untreatable?
Yeah,
so
that's
a
a great question.
And my sample, and you have the results.
Yes. And there's an ongoing discussion within the, our EAGJ group that whether we should give the participant the right to fully access their raw data.
Yes.
And our conclusion is that probably that person, if he or she wills, they have the right to access the raw data. But after undergoing another informed consent process, over the possible implications of the receiving the raw data. So that's what we are trying to do.
And as someone that does exome sequencing clinically, those kinds of things are not included when you do clinical exome sequencing. The data is there, but is just not returned to the patient.
Yeah, so that's my question. Can I ask to have those data?
So clinical companies like GDX, these other companies that do exome sequencing, will give data to patients if they request it.
Yeah.
One of the practical issues, which, it's sad that this is an issue, is that the cost of providing all the information is hugely greater than the cost of what is being done with the AGHI. And this is a real issue for this particular study that they really, one of the issues is what happens if 10 years from now, turns out that the medically actionable information has changed, and the interpretation of it has changed. What's the obligation to go back and find people now who may not be where they used to live, but you could put a big forensic effort out and eventually find them, but it would be expensive, and that's not part of the cost structure of the study as it's currently formulated. So there are all kinds of issues that are practical ones that come up that you prefer not to be part of the complications of the ethical components of this, but they're just ones that can't necessarily be dealt with. I'm not just throwing up my hands because maybe they have to be part of the way the study is structured. We should think about that.
So I'm not a clinician, but my question is, I see all of these calls, right, for participants, and I personally have friends who are researchers who need participants to do things, and I'm wondering, what's the ethics behind me participating in something very large, like say all of us, where I might actually one day be working with that data, potentially, probably not, but potentially, and I'm one of those samples, or my daughter is one of those samples, or my husband. What are because I feel like it's a little bit fuzzy, right? The ethics are a little fuzzy there.
Right. And actually, in the EJ participants contain clinicians and researchers as well. And yes, so, but one thing we can say that if an outside investigator wants to work on that data, they have to write up a IRB protocol, and it should be approved by the IRB before they can access to the data, the genomic data. So at that point, probably the conflict between self-interest and then access to one's own data may be screened.
Okay.
My question is for the medical treatable diseases, if they come back and they have breast cancer, they have the breast cancer gene, but they, number one, don't have a medical provider, or two, they don't have insurance, or they don't have the funds to get that treatment. Mm-hmm. What is the, I guess, the company's responsibility to helping the patients get the treatment now that they know that they have this medical disease or potentially do?
Yeah, that's
because in Alabama, I'm assuming that's a huge issue.
That's one of the issues we anticipate. And currently, we ask the participants to designate their, primary care medical healthcare provider, which we can discuss. So, but in the future, there may be participants who want to have the health insurance and medical provider in the first place. And we haven't come up with a solution yet, but we anticipate that at least if we can predict the genetic predisposition to the disease and encourage the participants to go to the screening, and that will eventually reduce the medical cost in the future by carefully monitoring the patients and prevent the disease. So that's what we hope, but we recognize that there is a question of future, how those kind of treatment can be covered by the medical insurance. That's an unsolved problem we have.
I think one of the barriers in Alabama is the low scientific literacy about this issue. I think there's a perception in the public that in medicine we understand very well links between single genes and diseases. And we're talking BRCA1, BRCA2, Huntington's, but a majority of diseases, multifactorial, probably many genes involved. So I suspect that the patients may be signing up thinking, I may find useful information medically, when actually our ability to deliver that promise is quite limited.
Yes. We try to, as part of our effort to mitigate the literacy, we have patient navigators who are not scientists, who come from the community they belong to, and they ask questions to us, and we feel the need of educating people more so that they understand what they are doing.
I'd like to just make you all aware that on Monday, March 18th, there will be a bioethics forum. It'll be held up in the Medical Towers building, which is on, I think it's 11th Avenue, where the Preventive Medicine Division is located, that will focus entirely on this issue of return of results from genetic studies in large population-based genomic studies. And it'll run from 9 to 1 o'clock or something, and it will include, we hope, some study participants who have gotten results that they found challenging and how that worked for them, ones who maybe have not, are waiting to get results and how they handle it, have some genetic counselors, have the patient navigators, and then bioethicists who are expert in thinking about these issues. And so I encourage you, if this is the kind of work you plan to go into or just it interests you, this is a real opportunity to talk about the issues, and we hope some sort of white paper will come out of that session, maybe not the session itself, but with some additional discussions to try to just make people more aware of what the key pressure points are and ways that we can be more fair in how we recruit people and how we set expectations for the studies. And as I think we all appreciate, it's an incredibly rapidly moving target. Just the fact that the list of actionable genes is different every time this presentation is given makes you realize that when you set up a study that's going to play out over several years. And the intent of this study, I guess, is to look out 10 years at least into the future of how the genomic data affects the healthcare outcomes for patients.
Here, recruitment will finish in 5 years, but the future research on the data will go on.
For quite a number more years. So this field will have advanced incredibly by the time the final phases of this study are going on. And how do you build responsiveness to that advancing of the field into a study where you've made commitments to patients at the start of what is going to happen to them and their information? It's very complicated and challenges everybody's best interests. So I didn't mean to cut off all discussion, but we'll continue to make people aware of programs like that are ongoing. That one will be available by the web if people want to participate from a distance. And we really encourage as diverse participation in the session as possible. It's time for a couple more questions if you still have any.